So it's the middle of finals season, and I definitely look like hell, but I wanted to talk to you about immunohistochemistry because even though it's not some cutting edge, revolutionary new discovery, it's still really cool, and not enough people know about it. First things first, let's talk about antibodies. Antibodies are these Y-shaped little suckers, and on each of these ends there's something called an antigen binding site. Great things about antibodies is that they're actually really picky, which basically means that an antibody will only bind to the antigen specified by its antigen binding site. At this point, someone had the very bright idea to use them as the microbiology equivalent of bloodhounds to hunt down a certain antigen in their sample. But how can you tell where the antibodies are? Well, easy. Tag them with something you can see. After a while, people figured out you can attach a fluorescent molecule called a fluorophore to the antibody, so now anytime the antigen is in your sample, it'll light up like a Christmas tree under the microscope. Magic. After that, there were a lot of cool developments that led to the more refined techniques that we use today. One of them is very popular and very simple in principle. It's called indirect or secondary immunofluorescence, and it basically involves two antibodies instead of one. Here's how it works. First off, you need to choose an antibody that'll bind to your target. We'll call this the primary antibody. You introduce it to your sample and let it bind to your target antigen, and then you rinse off the excess that isn't bound. Up till this point in the process, it's relatively the same as direct immunofluorescent staining, but for indirect you actually get a secondary antibody that'll bind to your first one. This is the antibody you stick a fluorophore on, and when you introduce it to your sample, it'll bind to your first antibody, which is already bound to your target antigen. And voila! It is now labeled. Now, this whole thing might seem kind of extra to you. I mean, why go through all the trouble of getting another antibody and doing the whole staining process twice when you could probably achieve the same thing with one? Well, I promise there's a point. The thing about direct immunofluorescent staining is that while the process has fewer steps, it also has lower sensitivity. You can only stick one fluorescent molecule tag per antigen in your sample, which means if your sample has low levels of the target antigen, it's going to be really hard to detect anything, and you have a higher chance of false negatives. With indirect immunofluorescent staining, you can actually amplify the fluorescent signal. This is possible because multiple secondary antibodies can bind to a single primary antibody, so you get multiple fluorescent tags per antigen, and that makes it a lot easier to detect in your sample. There are a few other variables to consider like cost and flexibility, but that's the gist of it. But what is immunohistochemistry used for, and why is it important? Well, it's an extremely powerful tool for diagnosing diseases, and it's actually frequently used to detect and diagnose certain types of cancer like lymphoma and mesothelioma. Outside of clinical applications, it's also widely used in research laboratories to observe and characterize changes in cells. That's all I have for you today, and I hope you enjoyed my little lesson on immunohistochemistry. Bye!